welcome him.
Good afternoon. Thank you for coming to my session. My name is Piotr Szul, and I will be talking about the genomic challenge and how we can use Spark to address it. And the challenge I'm referring to is looking for genomic underpinning of diseases and phenotypic traits. And this is important because knowing this, we can prevent and treat diseases. And it's challenging because human genome is quite long. It has 3 billion letters, and we are looking for only a few hundreds of places where the genome was disrupted. So it's kind of really like looking for needles in a large haystack, so hence the title. I came from Australia, from an organization called CSIRO, which is a government-funded organization for applied research. It's quite large, as you may see, and essentially it focuses on growing or fostering economy and well-being in Australia through scientific research. Data61 is an organization within CSIRO that focuses on data-driven future and covers the whole life cycle of data-driven approach for science and applications in technology and society. And if you haven't heard about CSIRO before, then probably you don't know, but you maybe should, that it's one of the birthplaces of Wi-Fi, and some of the very critical bits of algorithms and technology for Wi-Fi actually were developed in CSIRO in the early 1990s. Okay, so coming back to what I want to talk about, first of all, I wanted to give you a very short introduction to genome-wide association studies, which is one of the ways to look for these needles. Then I wanted to talk a little about variant Spark and cursed forest, which is our tool that we have developed to do this kind of studies, and then show you a few GWAS use cases. So how did we apply our tool to GWAS-like data sets? Oh, this one, okay. So genome association studies. So as I mentioned, this is one of the ways to look for those places in genome that can be related to diseases or phenotypic traits. And how do we do this? So essentially, let's say we want to look for a disease. So we find people that have the disease, and then we have people that don't have a disease. We get their DNA, we look at the differences and try to find places where they are statistically significantly different. And if we can identify this, we can say, okay, so these are the places which matter, this is where we should be looking for those genetic underpinnings. It's not as simple as it sounds. First of all, sequencing is still quite expensive, so we don't sequence the DNA. We use other technology called genotypic arrays, which essentially look at some specific positions in genome which are relatively commonly known to have variation. So we have about 500,000 of these positions called SNPs. And the entire workflow is also much more complicated because it involves a lot of data cleaning, quality checking. And even later, when we already know these positions, we need to still verify them, whether they have biological and clinical significance. So, but I will be focusing only on this part that deals with data and how you actually identify these associations having the data. So let's look at the data. And the data we have usually come in a form of one of those text formats that genomic people use quite often. And this can be, for example, VCF file, which is a variant call file. And this file, in every single line, has every single line identifies a single position in the genome and how different samples in our cohort or our study differ from some reference genome. And to look at this data and to analyze this, we need to first convert it to some numerical representation. And one of the ways to do this is just to look whether there is a variation or whether there's no variation, or more specifically, whether there's variation in 1 or 2 chromosomes, because as you know, we have 2 pairs of chromosomes for each position. So very simple encoding, 0, 1, and 2. And we end up in this very thin, maybe a few thousand columns, and very, very, very tall matrix of maybe a few millions of rows that describes our case. And then usually, when you do data science, we need to transpose it because it is common practice to keep samples in rows. And then we can try to look at the response variable, which in this case is categorical with 2 classes, and we can try to look at how or which predictors are mostly correlated with the response. And just to give you an idea of typical size of this study, so usually there will be about 1 million variants, maybe a bit more, maybe less, about a few thousand samples. But this is just because we look at those quite frequently varying variants, those SNPs, because the if we wanted to look at the entire genome and all the variation, then it'll be much more of those variants. So for example, we have this project called, oops, sorry, 10,000 genome project. And it shows us then that if we had a sample of about 2,500 people, then we have to look at about 80 million genomic positions where there's variations. Okay, and typically GWAS studies use univariate approach to look at the associations. So they essentially just would try to correlate every single position independently and then seek positions which are strongly correlated using some statistical test or approach. And typically, the results are presented in form of this kind of Manhattan graph that shows which positions are highly significantly correlated with the response variable. And if the significance is high enough, then we can conclude that these positions are likely to be relevant and likely to have some association with the disease that we are looking at. They are quite stringent, as you may notice. I'm not sure if that's feasible, but essentially the significance level for genome association studies are 10 to minus 8, so really, really small. And this is because we kind of test independently very many examples, so we need to make sure that we don't get random results here. And since 2008, there's been a lot of studies published, almost 3,000, and they have identified more than 30,000 different positions in genome that have associations with different traits and diseases. But there's a small problem here, and it's referred to as missing heritability. And heritability is a measure of how much of variant they observe in phenotype is caused by genomics factors. And for example, human height is one of the traits, and it's estimated that its heritability is about 80%. So 80% genomics or genome and 20% environment. But even though we have identified 40 different places in genome that can correspond to height, they only explain 5% of the variance. So there's a huge amount of variability missing, and it's kind of like dark matter of genomics. We know there should be more, but we can't see it at the moment. And the question is why? So there's a few possible explanations. One of them is that we need to run larger samples because we are not sensitive enough. Some of them are related to the fact that we are looking for only those common variants, so we are looking at only SNPs, and maybe there are some diseases that are caused by very rare variations, and we are not looking for them because of technology. But one of the explanations is that maybe it's because of the interaction between genes. And indeed, there is this concept called epistasis, which essentially means that genes interact together, and the expression of one or more genes can be regulated by other genes. And this comes in many different ways and forms simple Mendelian epistasis or something that may be polygenic, where multiple genes may contribute to continuous traits. So quite complicated possibilities of how that can interact. And this makes traditional approaches for modeling difficult. Because even if we wanted just to look at two interacting genes, we kind of square the size of the problem, running from, for example, 1,000 to hundreds of billions of possibilities that we need to study. So if not this, what can we do? And one of the ways people try to address this problem is try to use random forest. Random forest is a very smart machine learning technique developed in 2001 by Raymond. And very early on, researchers showed that it has capacity to discover interactions or to find them better than some of the simple statistical tests. So this is the result from the study published in 2004, which shows that the black bars are essentially higher than the white bars. It means that it's more powerful than the simple statistical tests. And why that would be the case? Why random forest would be actually a good choice for GWAS? Well, there's a few causes. One of them is that it's very flexible. It's not parametric, and it can model very, very complex interactions between variables. So it's quite possible that they can capture all the different combinations that the Epistasis can have. It's very resilient to uninformative predictors, which in GWAS is quite important because, as I said, we have those millions of different variables, only a few of them actually matter. So we need to be able to kind of not be distracted by them. And finally, it comes with the measure of variable importance, which can be used to identify variables which are contributing the most to the response. So these are the ones that we want to really identify. So, random forest. Okay, so one of the teams in CSIRO called Transformational Biology team, and by the way, this is not Australian accent, if you are wondering. Yeah, so we wanted to see if Spark can be used for large-scale genomic projects, and essentially if we can use Spark to apply machine learning in context of genomics. Spark because we have a lot of data, machine learning because there are some applications in which machine learning can be useful, like, for example, random forest. And the first thing we did just to try whether Spark is even applicable, we tried to just use simple k-mean clustering, and we realized that yes, it can be used. We were actually able to cluster the entire genome using our in-house cluster, and that gave us some confidence that Spark may be the way to go with machine learning on genomic scale. So then we went to random forest, and we ran into troubles. So even though our cluster is quite large, we were not able actually to run SparkML standard random forest on the entire genome. And the explanation for this, I think, was given in some ways last year on Spark Summit by, Faris Abuzaid, I hope I pronounced this properly. And one of the reasons is because random forest in Spark is optimized for data with many, many rows, but not too many columns. And essentially, the chart there shows that if the number of columns grows, the Spark becomes more and less efficient. And in some way, the communication overhead just essentially kills the performance. So the solution that he proposed was to partition data by columns, and that's what we independently decided to do as well. So we came with this implementation of random forest, which we call Kerr's forest, as if addressing Kerr's of dimensionality. And essentially, it's a very simple idea. Instead of partitioning data by rows, we partition data by columns. In our case, columns on those samples of size may be 1,000 or a few thousand of things. So it's quite easy to kind of partition them well. But also, it gives us a few advantages in terms of actual random forest algorithm. For example, we can technically, each executor can have all the data to find best splits, which are the core function of random forest algorithm. So essentially, this is a much more efficient way of dealing with this kind of data. And we implemented this in Scala. There's also some other tricks that make it faster. So for example, I mentioned these VCF files, which organize data also by column in some way. So we don't have to convert them, transpose them. We can just directly read them as they are and put them into our software. We realized that we can even make the communication even more efficient by building more trees at the same time, which gives us extra benefits. We really focused on optimizing the splitting process, which is the most frequent operation in random forest, so that it really works the fast on the data that we have. And then also we choose to represent data in a way that is most efficient for data style that we have. So what did we get with this? How fast did we actually are? And to give you some idea, I have run a simple test on a local machine, or one machine with 16 CPU cores. I'm comparing three things. I'm comparing SparkML random forest. I'm comparing Ranger, which is one of the fastest C++ optimized implementation of random forest, and then Kerr's forest. And as you may see, at least in this setting, this is a data sets of about 1,000 samples and from 100 to 1 million different variables. Our implementation is comparable, or maybe a bit faster, or at least in the same scale as the Ranger one, and it's 10 times faster than Spark native implementation of random forest. Yes, so it's fast. And then let's look at a more bigger data set, and this is actually showing the time to build 100,000 trees. And I'll explain why 100,000 in a moment. But the first conclusion is that we actually were able to build random forest on a data set with 50 million variables and 10,000 samples, so pretty much the size of the entire genome of what that would be. And then we look at this time to build 100,000 trees. So in the setting of GWAS, because there is so many informative predictors, we need to build many trees. And 100,000 seems to be a good number, so quite a lot of them. And if we look at the time to actually do this, so for typical GWAS size of data, so a few million of variables and a few thousand samples, it takes between 5 to 50 hours to build the number of trees on the cluster of 16 nodes, so quite a small cluster. And I tried to see how much it would cost on AWS, and it's about $200 to get this random forest. So not too much. So this seemed quite feasible. If we went to the full genome scale, it becomes a bit more pricey, $8,000. And the time is also much longer, so it's about maybe a month. But this was built on only 180 executors, so not a large cluster, and there is potential to scale it up, use more nodes, and reduce the number of time they actually need to get the same number of trees. Some other features that we have. Oops, what happened here? Okay, sorry. We have various input formats. We have lots of different random forest tuning parameters. And one of the distinguishing features is that we can look inside the model more deeply than other implementations. And we can, for example, extract the importance per single tree. And this lets us later to run statistical analysis and, for example, use bootstrapping to estimate better the importance of particular variables. That's what is shown here. OK, so now I wanted to talk about two applications or two cases where actually I tried to use this to identify some of these hidden variables. So one of them was simulated data study in which we generated artificial data set of 2 million variables and 5,000 samples and introduced 5 informative variables into it with varying weights, or kind of decreasing weights. And the expectation was that if we run random forest and try to look at the importance, we should be able to see that the importance is pretty much reflecting the weights of these variables. To measure this, we use a metric called rank bias overlap, which essentially compares rankings, and we focus on the initial variables. And the results are showing, and there's the little scale there that shows essentially what the RBO will be for different things, for quite wide range of parameters and number of trees, we're able to recover at least 2 variables, and the best model were able to recover 4 of them perfectly. So 4 of them were actually of the first one, and the 5th was a little bit behind. So this is quite a good result, even with quite a lot of noise, because the model was quite noisy. We were actually able to find what we were looking for in this simulated example. It is very basic, but it shows that it essentially works. So the next step was to look at some real example. And for this, we chose to use an existing study that tries to find correlations between genes and bone mineral density. And bone mineral density is one of the factors that can be used to measure risk of osteoporotic fractures. And I just wanted to kind of show you this slide to emphasize that these are very important studies, and they really may have impact on society, because these are really important or very nasty diseases, if you will. So the original study was done on not the big sample, so about 2,000 samples and a few 100,000 of SNPs. And essentially, the results were that they managed to replicate 21 of known genes that are associated with BMD. They identified 2 new ones that are highly correlated, and then they maybe found 4 more. And we essentially what we did just took the same data and run random forest on our cursed forest on this data and try to see what the importance of the variables would tell us about the same study. And the results are visualized here. And essentially what we have here are all the genes that they found, plus 2 others. And the x-axis shows us importance, and the more important things are on the left-hand side. So essentially, these are the most important thing. And the first thing we did, we ran a statistical test that confirmed that our ranking is significant. It means that the locations that they identified are ranked much higher than those that they didn't identify, which is good because on overall means that it's working. And the other thing that we found out is that there are two locations that were very highly ranked in our situation based on importance and which were not mentioned in their study. And we were able to come with some possible explanation why that would be, why they might actually contribute to BMD. And one of them, I think collect 10 was actually later confirmed by other papers as being actually related, which is good. And then lastly, one of the positions or gene that they were not able to replicate, the DC5, was ranked very, very low on our list, and later was disproved, or essentially other studies showed that it's not related. So I guess the conclusion is that to great extent, this looks like a promising working way to identify this location because it on overall confirmed the result. It finds some new things that were not in this study but were confirmed later, and it shows that some things that were not confirmed are also not confirmed here. In terms of the future, we kind of want to extend this in a few different directions. One of them is to try to implement on the platform so that people can actually use it and try it on their data sets. The other thing is we try to add more algorithms and integrate it with other genomic platforms. And I've heard the talk about Hail recently, and it sounds like a quite good place to start. And then also try to run more case studies using other GWAS data. And specifically, there is one algorithm here that I think is quite promising, which is called gradient boosted trees, which was shown in some research to be possibly better than random forest for identifying interactions between variables, and which is much more difficult to scale in traditional HPC. But I think that with Spark, it actually is going to be feasible to scale it on the big data sets. So just a few references, everything that I used to create this presentation. And conclusions are essentially that Spark seems like a feasible platform for machine learning on the population scale genomics data. That what we developed, the cursed forest, is a promising alternative for the GWAS. And finally, more technical insight is that data size and shape and type really matter, and we really need to look at how I'm going to optimize and possibly come with implementation for different data sets. Variant Spark is available as an open source project on GitHub at this location. If you want to give it a try, please do. Thank you.
Okay, thank you very much, Piotr. Any questions for him, please? Yeah, great. Thank you.
Hi. So does the random forest algorithm work for correlated data, like if you have family data?
It does, although...
Or is it going to be biased?
Sorry?
Is it going to be biased because you have clusters? Sorry.
This is a very good question. The answer is it will work, although maybe the results are not going to be as good as with the correlated data. Actually,
Short and succinct. So, any other questions? Oh, we've got some over here. I'll come back to you. Okay. Thank you.
So are you planning on implementing this algorithm and maybe porting it to the general Apache Spark code base at one point? Or at least maybe like try to do a few PRs that might make wide random forest more feasible?
Sorry?
Or at least maybe have some PRs that would kind of use your implement some
PRs, sorry.
Yeah, like for pull requests.
Pull requests. Yeah, so as I mentioned, there isn't this thing called Yggdrasil, which is implementation that was proposed last year, which deals with similar problems. So at the moment, we are really trying to optimize this for GWAS, and because of this, it doesn't really fit too well into the pipelines in the way Spark works at the moment. So I'm planning to look at Yggdrasil and see whether we can try to combine these things together. And if it's possible, then yes, then I might try to make a pull request later.
I was just curious about your challenges that you had going from traditional HPC to Spark.
When I mentioned traditional HPC, it was more trying to say that random forest is very easily scalable HPC because tree can build independently, which means that you can add machines and essentially build much more, many trees just by adding more machines. And but this only works for random forest. It doesn't work for these gradient boosted trees where trees are dependent. We essentially developed this on Spark without using HPC as such, so we didn't face any challenges here.
Okay, any further questions? Okay, oh, one more, okay, great. Thank you.
How deep in general were your trees? I noticed the fairly lengthy time for training, and obviously with such a wide forest, but were these trees very narrow or quite deep? Well,
the trees the depth was not limited, but they were limited by the sample size. So if you have thousands of elements to build a tree, the tree can be only 1,000 levels deep, or potentially maybe log 2 of this, so about 30 levels or something like this. So they are not very, very deep because the thing you split is the sample size, so thousands of elements, not billions.
Okay.
Okay, great. Thank you very much. Please show your appreciation. Thank you.
Thank you.